Hello, welcome to our webinar on meeting the demand of increasing NGS capacity with high throughput automation, a collaboration between the Center for Genomics and Bioinformatics, Beckman Coulter, and Illumina. I'm Alisa Jackson with Beckman Coulter, and I will be the moderator. We're pleased to bring you this panel presentation by Zack Smith from the Center for Genomics and Bioinformatics. He is a project scientist. Mary Blair, a senior application scientist with Beckman Coulter, and Dave Delano, a marketing manager from Illumina. A few housekeeping items before we begin. The presentation will run about 40 to 45 minutes. Then we will follow it with a question and answer session that will be moderated by myself. You can enter questions throughout the presentation by clicking on the green button on the bottom left-hand side. And you can also increase the screen size by clicking on the little frame icon that's located on the far right. After the presentation, please feel free to visit Beckman Coulter's booth, where we will continue chat around this topic and others, and you can download information about our genomic products and solutions. With that, I will get on with the presentation, and I will turn this over to Zack Smith. Zack?
Hello, Alisa. Thank you for coming to this presentation. My name is Zack Smith. I'm a project scientist with the Center for Genomics and Bioinformatics at Indiana University. I'd like to start by just going through our agenda real briefly. First, we'll discuss the transcriptome and RNASeq and why it's important. And then I will pass it off to Dave Delano from Illumina to discuss their RNA stranded kits. And Mary Blair from Beckman will then discuss about the development of this automation protocol. And then finally, I'll go through the QC data that we've generated as a part of this over both the HiSeq and MiSeq Illumina platforms. So the transcriptome is extraordinarily important. It represents the summation of all RNA species that play a role in the transmission and attenuation of information being passed from the genome to the proteome. It includes a wide variety of RNA species, including standard mRNAs, as well as small RNAs, microRNAs, and non-coding RNAs of various lengths. We are just now beginning to understand how increasingly important, how important the transcriptome is to really understand genomics in the modern times. The transcriptome drives huge numbers of changes in organisms. So a couple of things that we look at here at our institute is changes in response to environmental stimuli. So on your left, you'll see a couple of the common water flea, Daphne pulex. And the flea on the far left has been reared under standard laboratory conditions. It looks quite transparent. And the flea on the right is very red. It was raised in a hypoxic environment, and so is overexpressing hemoglobin to compensate for the low oxygen in the water. This is a fairly straightforward shift that you can see with the eye, but there are hundreds of other genes that are being upregulated and downregulated as a result of just this one simple environmental stimuli. And what we would like to know as researchers is, under other circumstances such as increased metals and acid stress, what genes are we seeing being transcribed in the organism? From a medical standpoint, the transcriptome can be used to observe shifts between cancerous and non-cancerous cell fates. And so that's extraordinarily important in oncogene research. So traditionally, RNASeq has been sort of blind to strand information. So during the library construction of traditional RNASeq libraries, the strand information is lost. And that's a problem because it leaves us with an incomplete picture of the transcriptome by ignoring a number of things like antisense expression. And it also does not allow us to be as accurate as we can be in looking for measurements of overlapping genes and looking for splice junctions. So if we can preserve the strand information during library construction, that actually gives us a higher percentage of uniquely alignable reads, which reduce your sample costs, actually increases the value of your experiment by maximizing your return on your investment. Additionally, there are some papers, including the ENCODE and ModENCODE projects, that seem to think that something like 75% or more of a genome might be transcribed at some point. In the water flea Daphnia, we have identified, in addition to about 38,000 genes, large numbers of what we're referring to as transcriptionally active regions, which show variations in transcription on application of metals and such. So the transcriptome is very important, and getting that strand information is also very important. Another reason why strand information is important is the model system that you're working in. So these are three very familiar friends in evolutionary biology. There's Drosophila melanogaster, the fruit fly, the watercress, Arabidopsis thaliana, and the comet lab mouse. These are systems that have been worked on in biology for many, many years now. Have very highly they have well annotated genomes for and for model systems, and have been very heavily worked on. These are good workhorse organisms for doing really good biology in. However, here at IU, we have a number of researchers who are trying to answer some very interesting biology questions with non-model systems. So you can see there the little bird is the junco. So we have a lab on campus that's using that as a model for social interactions. We have the sea urchin Heliosideris, which is on the bottom left. Orthophagus taurus, the horn beetle, and ferns, and all kinds of plants that we're working on, including strawberries and avocados. Systems where there are not very well developed reference genomes. And so if we can maximize the return on our transcriptome research by getting very accurate mappings of splice junctions and such, we can leverage the somewhat underrepresented reference genomes as well as we can. So it's very important for a non-model system. Additionally, the ever-increasing capacity of Illumina sequencers are tempting our researchers towards larger and larger studies. We have labs on campus that are interested in looking at entire populations of animals on a transcriptome level, as a result, looking at impacts and environmental stresses on whole populations. So that will give you a lot of samples to prep. We have other researchers who are interested in time course experiments, say following the activation of genes in plants after pollination. So looking at 1 hour intervals after pollination. We have other researchers who are interested in looking and actually getting to the point where we can do replicates of our RNASeq experiments, just to verify that the variations that we're seeing in the expression are not driven by library construction, that are actually biologically relevant. And so all of these expansions result in more samples, more libraries to prep, and they will really ideally benefit from decreases in cost in both time and money for library construction. So that's what's really pushing us towards the automation of these stranded kits. And so with that, I'm going to pass control to Dave Delano, our Illumina representative.
Thank you, Zach. I appreciate it, and it's my pleasure to be here today to talk to all of you about TruSeq stranded mRNA sample preparation kits. So our goal in designing these kits was to provide the clearest and most complete picture of the transcriptome. We went about achieving this by leveraging our existing core TruSeq RNA chemistry and the high coverage uniformity that it provides, and adding to that very precise strand information. And I'm going to talk in a minute about how we went about achieving that. We also further streamlined the workflow and added greater flexibility to fit a wide range of study designs with kit configuration suited for both low sample number and high sample number study designs. So how do we capture strand information? Well, many of you may be familiar with the study entitled Comprehensive Comparative Analysis of Strand-Specific RNA Sequencing Methods. So this is published a couple years ago in the journal Nature Methods by a group over at the Broad Institute. And this group compared 7 strand-specific RNA library prep protocols looking at multiple categories related to data quality, including strand specificity, the evenness and the continuity of transcript coverage, as well as the coverage of 5' and 3' ends, which, as many of you know, can be a challenge to capture. So this group ultimately selected fragmentation random hexamer-based DUTP as their top method, stating that the DUTP method provided the most compelling overall balance of criteria. So Illumina basically further optimized this protocol by improving the percent strandedness and also shortening the total assay time. So how does the DUTP method work? Well, the first step is RNA fragmentation. So for those of you who are familiar with working with RNA, you know that it tends to form secondary structure. So the goal here is to maximize the fraction of that transcriptome sequence that's going to be available as a substrate for cDNA synthesis. The next step is going to be first-strand cDNA synthesis. This is going to be driven by random hexamers. And the purpose of that is to maximize that uniformity of coverage across the transcriptome. Once that step is complete, you then go to second-strand cDNA synthesis. And this is, again, driven by random hexamers to bolster that coverage uniformity. Now, with this step, you're going to swap out the dTTP in the nucleotide mix with dUTP. And the reason for that will be apparent in a second. So your next step is going to be to ligate your adapters. And for those of you who are familiar with our chemistry, you know that we have two adapters, the P5 and the P7 adapters, which are specific to the 5' and 3' ends of your inserts, respectively. So once that step is done, you want to enrich for the successfully ligated inserts through PCR amplification. So with this step, we use a high fidelity TAC, and that TAC is unable to process through DUTP incorporated template. As a result of that, only one of those two templates is going to successfully amplify. And because you know the orientation of the P5 and P7 adapters relative to that original RNA transcript, you've now captured strand information. So I also mentioned that TruSeq's fragmentation random hexamer chemistry provides high uniformity of coverage, and that point is illustrated in the figure below. So if you look at the bottom left, this is based on a library generated using the TruSeq RNA chemistry. And on the x-axis, you're looking at the transcript from the 5' end on the left side to the 3' end on the right. And on the y-axis, you're looking at the level of coverage that's provided. And what you can basically see here is you're getting extremely even coverage across that whole span from the 5' to the 3' end. Now, in contrast to that, if you look to the right side, this is a similar experiment run with an alternative method. And what you can see here is that rather than seeing a straight line indicating very uniform coverage, what you're seeing is peaks and valleys with very high coverage on the far left side and the 5' end, as well as on the far right and the 3' end, but you have some gaps in between. So the repercussions on the experimental design and the quality of the study are that if you're looking for such features as alternative transcripts or gene fusions, the likelihood of your successfully and with high confidence capturing that information and making those calls is going to depend in part on where within the sequence those features happen to lie. And the only way around that is to increase the overall output, the overall sequencing output per sample, which may ultimately mean fewer samples that you can run per lane. So overall, because you'll be bolstering coverage in regions where it's not necessary, this means a less cost-efficient experiment. So in short, the higher that coverage uniformity, the more cost-efficient your study is going to be. So this slide is just showing the different configurations we have available for the TruSeq stranded mRNA kits. There are two LT or low throughput configurations, set A and set B. Each of these is going to give you 12 of 24 total available indexes. We also offer an HT or high throughput kit. So this is designed for higher throughput, larger sample number studies. It gives you 96 samples and offers you 96 indexes. So this is going to give you a little bit more flexibility in terms of the number of indexes you could run per lane. I also wanted to take a second, to plug in an additional kit in our portfolio that's related to that I just described, called the TruSeq Stranded Total RNA Sample Preparation Kits. So these kits are based on the same core chemistry of the kit that I mentioned, with the exception that they have integrated ribosomal RNA reduction chemistry, which enables the capture of both coding and non-coding RNA. Very importantly, this chemistry is robust with low quality, including FFPE samples. And there are multiple configurations available that will suit a wide range of study designs, including for specific species, as well as a kit designed specifically for blood-derived RNA. I also want to mention that I have it under advisement that Beckman will soon be offering an automation solution for these kit configurations as well. Now with that, I will pass it back over, to Mary Blair.
Thanks, Dave. Now we're going to switch gears a little bit and talk about why you would want to automate your library construction process. As we all know, good library construction is essential for quality sequencing results. By automating library construction, it allows you to standardize your procedure, free up your time for data analysis, increase your throughput while achieving those same quality results, reduce opportunities for cross-contamination, and protect users from handling hazardous materials such as samples or reagents. Now we can discuss what you can consider when you start automating your process. Everybody handles automation a little differently, but there's a particular process I like to follow. First, I review the protocol and start to map out the process so that I can highlight any incubation times or temperatures that are required, and any volumes that are being transferred, all the various reagent types that are used, and any start and stop points within a protocol that allow for partial processing of samples safely. From there, I look at which platform I want to automate on. Beckman Coulter offers 3 different levels of automation, starting with the Biomic 4000 for lower throughput, a Biomic NXP for medium throughput, and a Biomic FXP for higher throughput. Usually I'm given a throughput requirement and I choose the platform that meets that. However, I can also be given a platform and then determine what the throughput is capable of meeting on that particular platform with that particular application. While I'm looking at platforms, I also need to decide if there are any devices that need to be integrated. Beckman Coulter has many different options for integrations to handle your heating and cooling, any thermocycling, shaking, or specialty things like vacuum filtration and centrifugation. After I'm deciding on the platform and devices to integrate, I can start configuring a deck layout. For the Illumina applications, we chose to use the Biomic FXP Dual Hybrid in order to get 96 samples with minimal amount of user interaction. We can also use the Biomic NXP and the Biomic 4000, but we would have to use a lower throughput requirement in order to use those. Choosing the Biomic FXP allowed us to use the 4x3 Alp so that we have a max amount of space for labware, a 96 tip wash station so that we can cut down on consumable costs and wash our tips during all the ampere cleanup steps, a static Peltier to keep the reagents chilled, an orbital shaker to cut down the overall processing time by eliminating some of the tip mixing, and to have space for the optional T-robot so that it can be a more walkaway solution with having your incubations handled on deck. Zack and I worked together to build the Biomic method, and we did this by outlining the protocol within Biomic software. Biomic software allows you to easily build a method by using these drag and drop GUI icons that you can configure to meet your protocol. After we were finished building the Biomic method, we started to do water tests so that we can get the initial pipetting optimization complete, get the method flow down, check all our labware definitions and our labware moves on the system. Then we can do pipetting optimization using like reagents. I like to use glycerol solutions to mimic master mixes, or if I have any available, use expired reagents. Doing this allows me to cut down on my developmental costs. After we're done with pipetting optimization, we start our live sample runs. We started working with a small sample batch, such as 8 samples, and then worked our way up to doing the full 96 samples. Doing this allows us to see if there's any column effects that are taking place between a small sample batch and a large sample batch. And for the Illumina applications, I applied a basic user interface using HTML. And I don't always do this for all of my applications. If it's a static process or a single customer, I will typically tend to skip this part. There are some things you need to consider when moving from a manual to an automated process, and there are some challenges that can happen, but we can easily manage those. For the Illumina application, we had to realize that labware selection can affect the quality of results. We chose to use a half-deep 96-volt plate for our ampere cleanup steps because this has allowed us to use shaking instead of tip mixing, which cut down time and made it more consistent. We also used the Biorad PCR hard shell plates because these plates are very automation friendly and allow us to move the plates on the deck and in and out of a T-robot so that we can incubate without having to have user interaction. For the static Peltier, we developed our own tube block so that we could keep the reagents chilled. And we also chose to use a V-bottom shaped tube because this allows us to cut out on the amount of dead volume so that we can keep our reagent costs down low. For other reagents such as ethanol, when we were more concerned with evaporation and its hygroscopic nature, we actually used an overage amount in order to compensate for those. Overall, the hardest thing is actually pinpointing all the little nuances that people do on a manual process and then translating those to an automated process. I've worked with some chemistry in the past where the brand of water had a great effect on the efficacy of the kit. However, in this instance, the Illumina chemistry was actually very robust and very easy to work with. So why did we develop the Illumina TruSeq stranded mRNA method suite? Well, we wanted to help you increase your throughput, reduce errors, and help streamline the workflow. The suite of method includes the cDNA synthesis and library construction method, an Ampere XP cleanup, kappa qPCR setup method, and the normalization and pulling method. And each one of these is capable of processing up to 96 samples. The first method is actually two methods in one. And this is where you prepare your libraries. It allows you to select a kit so you can use either the TruSeq stranded mRNA kit or the TruSeq RNA version 2 kit. You can also enter your number of samples anywhere from 1 to 96, choose whether you want to incubate on deck or off deck, and then if you want to run cDNA synthesis or library construction. Here we have cDNA synthesis selected, and so we can choose to perform poly-A purification and fragmentation through second strand synthesis, or if we uncheck the perform poly-A purification, we can now bring already purified mRNA onto the system. If we change the method that we're running to library construction, it also updates the options. So if we choose to run the TruSeq RNA version 2 kit, we now have the option to perform end repair. We still have the ability to choose the number of samples we're processing and how we're handling our incubations. There's also some options that are included for the adapter. We allow you to pause for addition of the adapter labware to the deck in order to prevent evaporation of your adapters. You can also select which labware type you're going to use. You can use the Illumina wrap plate or a customer-defined 96-well plate or 24-position tube rack. And you can use a transfer by file option in order to hit pick. We also allow you to alter the volume of sample being used in your PCR setup. The second method is also two methods in one, where it runs Ampere XP and your qPCR setup. However, this one's a little bit different than your library prep method. This one you can either run each method separately, or you can run them together in order to minimize the amount of user interaction with the system. The user interface for Ampere XP has some options for the number of samples that you're cleaning up, your PCR sample volume that you're using, the volume of Ampere XP, and how much you want to elute in. The qPCR setup user interface has many options available as well. You can choose between doing a 96-well format or a 384-well qPCR plate format, how you want to handle your dilution plates, and how you want to set up your qPCR plate. We've done all our testing using the Kappa Biosystems library quant kits. However, we can actually alter this method to work with any other qPCR kit type if requested. The last method is the normalization and pulling method. And in this one, again, you have to enter the number of samples anywhere from 1 to 96, and you also input the options that you used for your qPCR and the average fragment size you want to use so that you can get a more accurate quant. And you also select whether or not you want to run pooling. The reason we need to know your qPCR setup is that the normalization pooling, the first thing it does, it pops up a spreadsheet. And this spreadsheet is very simple to use. There's 3 basic steps. 1, you import your raw qPCR data. 2, you enter the volume and concentration that you want to normalize to. And 3, you click the Export Data to Biomic button. But the spreadsheet actually has a lot more that's going on here, and it's kind of hard to show in this webinar. There's a separate tab that handles your pooling, and you can either hard code your pooling options if you're running the same process each time, or if you want to change your pooling volumes and destination wells, it actually allows you to set this each time you run. There's also some error checking that's built in, as you can see from the red text. If anyone wants more information on the normalization method or any of the methods that I've talked about, please feel free to go to the Beckman Coulter booth after this talk, and I'll be hanging around so that I can answer any questions. Finally, you can see the timing for the instrument when running the Illumina stranded mRNA kit here. Zack and I broke it down into a 2-day process, where day 1 was cDNA synthesis, and it took about 5 hours to run 96 samples. Day 2 was our library construction day, and it took about 4 hours to run 96 samples. And now for the more important part of our presentation, I'm going to pass it back to Zack so he can talk about the data that we generated using this Illumina method suite.
Thank you very much, Mary. So as part of our quality control of this automation method, we did 2 separate sequencing runs. One, a 24-sample library construction run that was run on the MiSeq here locally at the CGB. And then we also did a 96-sample library construction run, of which 30 samples were sequenced at Illumina's facility on a HiSeq 2000 instrument. So I'm going to now just dive into the MiSeq data. So for the 24-sample MiSeq test, we ran mostly Daphnia pulex RNA samples. So the plate layout during library construction, you can see on your left in this slide. There's a large number of Daphnia samples. There are human universal reference RNA samples from adjuvant at various concentrations running from 100 nanograms to 2.5 or to 2 micrograms of total RNA going into library construction. There's also a couple of Arabidopsis samples. The Daphnia and Arabidopsis RNA were provided by researchers here on campus. The plate was laid out in such a way that we could use the sequencing results to assess whether or not the automation was carrying samples over during the course of library construction. Humans, Daphnia, and Arabidopsis are quite divergent, and we are confident that we can tell the difference between those reads if we had, say, human reads that carried over into Daphnia samples and so on. So that's the logic behind our plate layout. At the end of library construction, we ran the libraries out on the adjuvant tape station 2200 instrument, which, if anyone's familiar with it, it's very similar to the adjuvant bioanalyzer, except it's more amenable to high throughput projects. And so you can see the electropherogram traces on the bottom right, and then false gel images of the library size distributions on just above that. And so the library results were very consistent in size and fairly consistent in concentrations at the end of library construction. So we were very happy with these results. So after sequencing, we sent the data for the universal human reference RNA libraries to our colleagues at Illumina, who have specially designed bioinformatics pipelines to use this information or to map these reads and to assess how well the reads are mapping to the human reference genome, which in this case is HD18. As you can see, our percent alignments reads in the second to the left-hand column are very high. We're getting over 88% alignment, with only about anywhere from 2 to 3% of the reads were unaligned. Abundant reads and were also quite low. Human ribosomal and ribosomal 5S subunit reads were extraordinarily low in this run, which indicates that the ribosomal removal that we employed during the course of library construction on this run worked quite well. And we saw very low percentages of mitochondrial DNA reads and adapter dimers. So we were very happy that these libraries are performing quite well. In these 2 charts, what the base counts in gigabases are on the left hand, are on the y-axis in both charts, for both the universal human reference libraries and for our Daphnia libraries. These graphs here do have also the in addition to the total bases, which are in red, the Q30 base, total bases were in blue and the Q30 bases in red. So the quality scores, or Q30 scores, are indicative of extraordinarily high confidence in the base calls of these libraries. And so what we're producing here are libraries that are very consistent in size, and the base calling is quite accurate. So we're very happy with these results as well. Locally, our bioinformaticians here at the Center for Genomics also went through the bother of mapping each of the libraries to the three different potential reference genomes on this plate: the human, Daphnia, and Arabidopsis. For human, we use the HD18 reference genome, we use the TARE10 reference genome for Arabidopsis, and we use the TCO genome for Daphnia pulex that's available on WFleabase. And so if you look at the data, we have the library on the far left, the total number of reads, and then the number of reads and the percent reads for each library that potentially might map to a different reference genome. So for example, in the Daphnia K13 library, we had 238 reads that mapped potentially to human. After these reads are very low from a total read standpoint. The percentages are quite minute. And on further investigation of these reads, we determined these to be mostly conserved sequences between genomes. So we're confident from the MiSeq data that we're not observing a cross-contamination problem. So those results encourage us to up the scale quite a bit. So on our second QC run, we did a full 96-well plate. Here's the plate layout. It's very colorful, but if you'll bear with me, I can explain it all. All of the red wells you see on this plate are tomato libraries. We have the universal human reference RNA libraries in yellow, Drosophila melanogaster in the peach. We have some Daphnia pulex libraries in blue, as well as Tribulbium libraries in purple. So it's a very mixed plate. Researchers here on campus are working on a wide variety of organisms, so this is a very good test for us. And again, the plate was laid out in such a way that we could use it to determine whether or not cross-contamination was an issue. So, again, at the end of library construction, we ran the finished libraries through the Agilent 2200 tape station unit, and our consistency of library prep was very high. We were extraordinarily pleased with these results. So average size was about 300 base pairs or so, which is good. Of the 96 libraries, 30 of them were selected for and sent to Illumina for sequencing on the HiSeq 2000 for 2 lanes of a 75-cycle paired-in run. And Illumina's pipeline was focused primarily on the universal human reference RNA libraries as before. So as you can see from this chart, we have a large number of, we're looking in the tens of millions of reads for each of these libraries that are passing their quality filters for both read 1 and read 2. And our percent filter pass reads with bases greater than Q30 scores are very high. So if you look on the y-axis, we're starting this at 82%, which is extraordinarily good. Our percent alignments for both read 1 and read 2 in the universal human reference libraries here are presented in red. We had high alignments, higher in read 1 than in read 2 because quality scores tend to dip a little bit during read 2. But on balance, these look really good. Our percent ribosomal reads were low, except for a couple of libraries where we observed some slightly higher percentages of ribosomal sequence, and we attribute this to a localized problem with library construction for those samples. Usually, the chemistry is very robust at removing ribosomal, or more correctly, at capturing the messenger RNA and moving it forward, and rather than leaving our ribosomal RNA around to be reverse transcribed. Our percent duplicates were very low for the universal human reference samples. They're somewhat higher for the tomatoes, but that's not terribly surprising given the repetitiveness in plant genomes. And our percent median CV coverage, so when we're looking at the coverage of the transcripts across the length of the of any particular transcript, was pretty close to 1. So this graph and another one I'll be showing later seem to indicate that we're not observing any bias towards the 5' or 3' ends. Very importantly, this is the percent correct strand orientation for each of the libraries. So specifically, we're going to, the entire goal of doing stranded RNA-seq is to preserve the stranded RNA, the strand information during the course of library construction. As you can see from the human libraries here on this samples, we had reads assigned to the correct strand over 98% of the time for these libraries, and that was we were extremely happy with these results. This shows that this library construction technique is actually doing what we wanted it to do. And again, this is a plot showing the distribution of reads over any particular position of a transcript. So as you can see, we're generating libraries that are not biased towards the 5' or 3' ends. Additionally, our colleagues at Illumina mapped their reads to to the human reference genome, and determined, not surprisingly, that the vast majority of our messenger RNA reads were coming from coding regions or untranslated regions or UTR regions. We see lower counts of reads coming from intronic or intergenic spaces inside the human genome, which is good. That's exactly what we were hoping to see. And then finally, as far as analyzing the 96 sample run, our bioinformaticians here at the CGB mapped each of the libraries to all the potential reference libraries on the plate. And so as you can see here, we have Drosophila, tomato, human, and tribolium libraries. Our Drosophila libraries mapped overwhelmingly to the Drosophila reference genome, as likewise for our human libraries. Our tomato and tribolium libraries did map also primarily to those genomes. The orange bars that you can see on these graphs are all unassigned reads. We don't really necessarily observe reads that are being mapped to the incorrect reference genome. The larger numbers of the larger percentages of unassigned reads in the tomato and tribolium libraries are more indicative of the current state of those reference genomes. So Drosophila and human are extraordinarily well worked on systems, and tribolium and tomato, the reference genomes just simply are not as good as for Drosophila and human. And so this is just a numbers presentation of that previous slide that I showed you, with the mappings of each species to its reference genome. So we have the species of the library, the total reads, and then each of the reference genomes that it was mapped to. So as you can see, we're not really observing significant numbers of reads that are mapping to incorrect reference genomes. So this would indicate that the library construction method is not causing sample cross-contamination during library construction. So with that, we can draw two major conclusions: that the automation of the stranded mRNA library prep kit from Illumina results in higher sample throughput with less time and less money, and that the libraries that we are constructing with this automation solution are very high quality and provide good results for sequencing while being free of cross-contamination. And with that, I'd like to acknowledge the hard work of our colleagues, Mary Blair and Alisa Jackson at Beckman Coulter, Tim Hill, David Delano, and Alejandro Gutierrez at Illumina, and my supervisor, Dr. Scott Michaels, and myself at the Center for Genomics and Bioinformatics. And so with that, I would like to pass the presentation back to our moderator, Alisa Jackson.
Hello, everyone, again. Thank you, Zach, Mary, and Dave, for a very interesting presentation. That was excellent. And hopefully everyone could hear me because I lost my connection there for a minute. We're going to go right into our Q&A session. And feel free to type in questions as we're going through this. It looks like we have a few questions. The first one is for Zack Smith, and that is, was the method developed in conjunction with IU, or did IU receive the already optimized method? In either case, how long did it take to get to a fully implemented method? And with that, I will pass it over to you, Zach.
Thank you, Alisa. IU did not receive any optimized method. We built this with Beckman Coulter from the ground up. It was advantageous that we didn't have to rework most of the chemistry in order to make it work on the automation. But between Mary Blair and myself, we built this automation solution from the ground up, and it took us a few weeks to get it up and running. There's a lot of advanced scripting that's involved in this method. And there's a lot of optimization that takes place when you're moving from a manual to an automated solution for library construction. You're just testing shaking times and making sure that you're delivering the correct volumes to perform what the chemistry is capable of doing. So yeah, we built this one from the ground up, and we're quite proud of it. So I'll pass it back to Alisa now.
Okay, Zach, I think this question is also for you, Zach, and Mary may want to add on to it, or I may add on to it after, depending on how you end up with your answer. What throughput justifies automation? And how many MiSeq customers have automated any type of NGS sample preparation? And with that, I'll turn it back over to you, Zach.
Thank you, Alisa. So in my mind, any amount of sample prep that exceeds 8 samples justifies automation. My facility is a small college or university core facility. We're limited on our staff, but we do have a large department doing lots of interesting research, and they are interested in turning their samples around quickly and reliably, and they want to make sure that they get the best investment for their money. So here at the CGB, we charge our customers based off of a sliding scale on the number of libraries that we're going to prep, because it does increase, if you're doing it manually, there's large amounts of increase in time largely, and technician time basically. So we try to automate anything that we're doing above 8 samples, simply as it is more cost effective to do it with automation than it is to do it manually when you're talking about anything beyond 8. As far as MiSeq customers are concerned, we have a number of labs on campus that are interested in bacterial sequencing, and bacterial genomes are small enough that library construction cost is really the biggest driver in determining how many samples they do. Because the genomes are small, you can easily get 1000x coverage if you're just talking about a single MiSeq run. And so for our microbial research community, they're very interested in prepping as many samples as they can as efficiently and cheaply as they can, even if they're only running it on a MiSeq. So I think that automation serves both the MiSeq and the HiSeq platforms very well. I think that given the quick turnaround time on the MiSeq, you can also do larger numbers of more complex organisms and eukaryotic systems. And so, yeah, I think it's a solution that works well for both platforms. And with that, I will turn it back over to Alisa.
Okay, Zach, thanks for that answer, and we're going to give you a workout here. The next question is for you also, and it's what percentage of barcode reads are perfect, and what do you do with the barcode reads that are one base pair off? And with that, I'll give it back to you, Zach.
Oh, thank you, Alisa. So for large numbers of multiplex samples, we really can't afford to have any ambiguity in the barcode base calling. And so when we're demultiplexing our libraries, we tend to, for reads where barcodes were not assigned, we tend to discard those. And those are a vanishingly small subset of any particular experiment, usually much less than 1%. Occasionally, what you can do with the MiSeq reporter software is you can add random bases if you're concerned that there's going to be particularly troublesome base calling events. But really, the best thing you can do as a researcher is to make sure that you're using barcode combinations that are complementary to each other and in accordance with Illumina's recommendations. Usually, the strongest problem that researchers have when we're doing multiplex samples is that we have index sequences that are too alike, which then result in the loss of cluster acquisition during sequencing. And so making sure that you understand what barcodes you're putting into any particular sequencing run, and hence any library construction run that you might do, is very important from a sample planning and from an experiment planning stage. And so with that, I will pass it back to Alisa, hoping that I have answered the question.
Okay, and we'll give a few seconds here in case anyone else wants to enter a question, or in case you wanted elaboration on any of the questions that were answered. And with that, it looks like no one is typing. So I would like to thank the audience for attending today. We really appreciate it. Again, I encourage you to visit our booth for some additional chat. You can download information on our genomic products and solutions for NGS and other applications like extraction for DNA and RNA, purification, size selection. Also, we have a survey going on in our booth, and if you fill out the survey, you'll be entered in a drawing for one of 2 iPads. So do stop by, and once again, thank you very much, and that's the end of our presentation.